How can we control, or how does a cell control, regulate enzymes? All right, so we're going to talk about a couple of different categories. We're going to talk about allosteric regulation, and that has activators and inhibitors. We're going to talk about competitive inhibition and feedback inhibition. So let's do allosteric regulation first. Allosteric. The prefix allo means other. So this is an allosteric site. This is an other site. Other than what? This is a site on an enzyme. Well, other than the active site. We know what an active site is. That's the site that binds where the substrate is. It's going to do the work. Allosteric is someplace else on that protein, on that enzyme. This is an allosteric site. So when we're talking about allosteric, we can have an allosteric inhibitor. So this is a molecule that binds at this allosteric site, so an allosterically, someplace other than the active site. But remember, we got this 3D conformation, and when you shove something in there, it's going to change the shape. And when that allosteric inhibitor binds, it changes the shape of the active site. You can see that represented in this picture there. So an allosteric inhibitor has changed the shape of the active site. Because it's considered an inhibitor, it's going to inhibit the active site. It's going to stop the active site. So in this case, this has shut down this enzyme's ability to work. It has a distorted active site. The substrate doesn't bind to it anymore. So this is considered allosteric inhibitor. This is when you send your cashiers on break. Send them to lunch. You know, you've got a break in the day. You want to shut down the enzyme. You're not telling them to go home, but you just want to stop it from working for the time period to conserve energy. This is allosteric inhibition. Okay, all right, so we can also have allosteric activation. Because we're talking about a site other than the active site, there are some enzymes that we only want to call them in when we need them. They get made, but their active site is non-functional. It's not capable, it doesn't fit to bind the substrate. So it's made with a non-functional active site. But sometimes we need some help. And in this case, you can have an allosteric activator. Allosteric, because it's going to bind somewhere else other besides the active site. And in this case, it's classified as an activator because when it binds, the active site changes shape and now it becomes functional. Now it's going to fit in the substrate. So when the allosteric activator is bound, I get a fully functional enzyme. You can get bound. This is when you call all available cashiers to the front. You know, you get a rush and you pull everybody off of whatever they were doing and bring them to there. You're turning on these enzymes so that they can get the job done and get through rush hour. That is an allosteric activator. Okay, so we have allosteric activation and allosteric inhibition, and both of these are allosteric methods of control. All right, so what else? What other ways do we know that bacteria regulate their enzymes? And many of these are true for eukaryotic cells as well. There's also a way of regulation called competitive inhibitors, so competitive inhibition. In this case, I have an enzyme, but I have a molecule that will bind in the active site. So instead of binding in the active site, and allosterically somewhere else, it actually binds in the active site and it keeps the substrate from binding. So this is a way you can kind of turn the enzyme off and make sure it's not working. If this molecule binds instead, it won't bind to the substrate from that. Okay, so this may be the coupon lady. This is somebody who's taking up space in the active site and keeping this enzyme from working. You know, I've got nothing against coupons, love me a good coupon, but you know, you're in line and suddenly the lady brings out the book and it's all this stuff and you are stuck. The enzyme is busy. You're never going to get out of Walmart. This is competitive inhibition. It's inhibiting, you're blocking it, it's stopping, inhibiting the enzyme from working, and it's competitive because it's competing for the active site. So it's blocking it in this method. So this is called competitive inhibition. Now this is reversible. The competitive inhibitor won't stay blocked, it'll come off and then the substrate can do that. And it's also a numbers game. If I have a lot of substrate, then my competitive inhibitor, just by numbers, isn't going to bind that often. If I had a bag full of marbles and there were 10 red ones and only one blue one, well, chances are you're going to get a red marble. You know, if there's a lot of substrate, then it's going to outcompete this inhibitor. Now, we take advantage of this method with our drugs. We can actually create a drug that's shaped like the substrate where it binds to the active site. We can create a drug that's designed to fit in that active site. And if that drug is put in the environment, it'll stop the enzyme from working, and we can shut down that pathway. Remember, it's a numbers game, so sometimes the substrate will win. So you have to make sure that there's enough drug. You have to increase the dosage. You have to make sure that the drug stays in the system, especially when we're talking about humans. You know, as soon as we put something in our body, it's our body's job to get rid of it. So the drug has to stay in a high enough concentration so it's successfully competing and shuts this down, or at least decreases it from that. But we use this a lot with drugs and bacteria. So this is competitive inhibition. All right, so the last one is called feedback inhibition. And this one starts with the diagram. We'll follow the diagram. This is our normal pathway down this side. So let's see what happens. We have a substrate. We have an enzyme. The enzyme binds. The substrate. You can see that locking key, that induced fit as we get the enzyme substrate complex. There is an allosteric site that's being pointed out here, but it's empty. It's not affecting it. The enzyme is functional without it. So this is what normally happens. The enzyme binds the substrate and converts it into an intermediate. So I have an intermediate A. There's another enzyme that binds to intermediate A, converts that into something else. I have a third enzyme that does that, and eventually get to an end product. Remember our representative pathway? That's what we just ran down. So I have an end product. But what happens if I have a lot of end product? If I have a lot of this, do I want to spend the energy to make more when I have a plenty, when I have a lot of end product? No, nobody wants to do that. Nobody wants to do work when it's already been done for them. So if we've got a stockpile of our end product here, we don't want to keep this pathway open. We want to tell it to stop. So this end product actually serves as a allosteric inhibitor. You can see it fits into that allosteric site. Okay, so that end product you can see fits into that allosteric site, and it changes the shape of the active site, and now the substrate can't bind anymore. So again, this is feedback inhibition. It's a way to control it, to save energy from this. It typically will target, remember when we target these different enzymes, that makes a difference too. Typically it targets an enzyme right at the beginning. Why? If I were to target an enzyme down here, well, I've done pretty much all the work, and we don't want to do that. So it's best with this. What we see with competitive inhibition is that it usually targets an enzyme right at the very beginning, first or second step in the pathway, and shuts it down. So all of those ways are regulating enzymes.